And with that said, it is my absolute pleasure to introduce our speaker today. So our speaker today is Dr. Dr Shorrock, who is a research scientist in the lab of Andy Berglund at the RNA Institute at the University at Albany. Dr. Shorrock received her Master of Science in Biomedical Sciences and Synthetic Organic Chemistry from University College London in 2014. In 2018, she graduated with a PhD from the University of Edinburgh, where she studied mechanisms of disease pathogenesis in spinal muscular atrophy. Following her PhD, Dr. Shorrock joined the lab of Dr. Laura Ranum at the University of Florida as a postdoctoral researcher. During this time, Dr. Shorrock transitioned from studying mechanisms of disease pathogenesis, or transitioned to studying mechanisms of disease pathogenesis in the CTG-CAG expansion disorder, Spinocerebellar Ataxia type 8. Dr. Shorrock joined the Berglund Laboratory in 2020, where she studies shared mechanisms of disease pathogenesis and small molecule therapy development across different kinds of spinocerebellar ataxias caused by CAG repeat expansions. Dr. Shorrock has also been a writer and editor for SCAsource since 2018. And with that, thank you so much for joining us today, Dr. Shorrock. We're excited to hear your presentation. The floor is yours.
Well, thank you very much for that lovely introduction, Celeste. Thank you all for coming to join us. And hopefully I'll be able to provide you with a nice overview of some of the what we think of as the basic biology of SCA8. I'll be able to tell you about the model systems that we use to model this disease in the lab, as well as talking about the current status of therapy development and clinical trials in SCA8. And then finally, I'll move on to discussing some of the current research in SCA8, and in particular in association with something called reduced penetrance of SCA8. So let's start with Spinocerebellar Ataxia type 8 is caused by a repeat expansion mutation. And so what we have in SCA8 is we have this repeat expansion, and this occurs actually in two genes that sit on opposite strands of the DNA from each other. And so in the ataxin 8OS gene, we actually have a CTG repeat expansion associated with SCA8. And this is also in the gene on the opposite strand of the DNA, ataxin 8, where that is a CAG repeat expansion. And so these repeats would normally be in the range of 15 to 34. So most individuals will have a variable number of repeats here, but it's generally going to be less than 34 repeats. Now, I don't want you to spend too much attention on the specific numbers here, and I'll come back to that a little bit later on as to why. But in general, when you get kind of over about 50 to 80 repeats, we start to see these repeat expansions being associated with Spinocerebellar Ataxia type 8. And so we can think of SCA8 as being caused by a CTG-CAG repeat expansion in the ataxin 8OS, ataxin 8 genes. And so sometimes you might hear to it referred to as a CTG repeat disease, and other times you might hear to it referred as a CAG repeat disease. Now, this repeat expansion is inherited in a dominant manner. And so if the disease presented in a dominant manner, what that would mean was, you would inherit a CTG repeat expansion, let's say, from your parent, and both parent and child with that repeat expansion would present with the SCA8 disease symptoms. Now, that isn't the case for SCA8, because SCA8 has reduced penetrance. So that might mean that there might be multiple individuals in a family that have this CTG repeat expansion that is inherited dominantly, but not everybody with that repeat expansion will actually develop the disease. And so we think of SCA8 as being a dominant spinocerebellar ataxia, but with reduced penetrance. And I'm going to come back to that idea later on. Something else that's quite interesting about SCA8 is that it actually has a really variable age of onset. So we have cases that present with spinocerebellar ataxia with ages of onset ranging from birth to individuals up to 79 years of age. Generally, SCA8 is a slowly progressive cerebellar ataxia, and so that ataxia is really the loss of balance and coordination. It comes with other symptoms as well, such as slurred speech and alterations in eye movements. And this is all really driven by degeneration of this region here of the brain called the cerebellum. And there are also effects from the brainstem, which is this region here that connects your brain to your spinal cord. It's a particular type of neurons within the cerebellum called Purkinje neurons, which are really big, really large neurons that have connections all over the brain. And so these neurons are some of the ones that really degenerate in SCA8 and cause a lot of the problems. And so we'll think about those neurons in particular later on. So I'm now going to transition to talking about how we can study Spinocerebellar Ataxia type 8. So in the lab, we need model systems that will allow us to be able to investigate why these mutations are causing the disease symptoms, and to start to think about how we can develop therapies to prevent that process from happening. So to do that, we need model systems, and these need to be really disease-relevant systems. And so for SCA8, we have a really wonderful transgenic mouse model that demonstrates various symptoms. So these mice show alterations in their movement and their balance, and that's something that we can measure in the laboratory to see how they're moving and how they're behaving. And we can use that as a readout if we're doing therapeutic studies, for example, to assess if any of the treatments that we're using are improving those behaviors in the mice. The other thing that we can use these mouse models to do is to understand more about the disease process itself at the molecular level. So what is happening in these cells during the disease when they express this repeat expansion? And so one really key finding that was found from this mouse model was actually the presence of these proteins. So this is a Purkinje neuron here, and you can see in the nucleus, you've got this bright red dot, and you can see that in all of these cells along here. So this is within the cerebellum, and you can also see it in these neurons in the brainstem here, these bright red dots. When we look in the brains of control mice, you can see nice healthy neurons here. So that's one neuron, and here's another. And none of these neurons, or any of the neurons in the cerebellum, in the brainstem, sorry, had these really dark red dots. And so this actually taught us that in SCA8, we actually get disease proteins that are made from the ataxin-8 gene. And so this finding was published in 2006, which is 7 years after the original publication that reported SCA8 to be caused by a CTG repeat expansion in the ataxin-8 gene. And so it wasn't until we made the mouse model that we actually understood that this CTG repeat expansion in ataxin-8 is also a CAG expansion in the ataxin-8 gene on the opposite strand. And it's from that ataxin-8 gene that these proteins are made. And we're going to keep thinking about these disease proteins throughout this talk. And so one of the other ways that we can study these proteins is using transgenic cell systems. So what we can do with very easy to grow cells in the labs that are kind of very standard, simple to use system that are really readily available. We can grow these normal cells in the lab, and then we can do something called a DNA transfection. And so this can introduce a bit of DNA that we're interested in studying the effects of. So in this case, we're going to transfect this cell with the ataxin-8 repeat expansion. And so through this DNA transfection, we get this ataxin-8 DNA into the cell, and that produces the ataxin-8 expansion RNA. And that leads to the production of these SCA8 disease proteins here. And so what we've done with this DNA transfection is we've turned these control cells actually into a cell model of SCA8. And so with that model system, we can understand exactly what these cells are doing, these proteins are doing within the cell. We can start to understand what the RNA might be doing in the cell and how this affects the overall cell health. Now, what's really, really nice about when we think about the SCA8 transgenic mouse model system that I mentioned, as well as these transgenic cell systems that we can do really readily in the lab, is that we can look at similar things across multiple different model systems. And so in science, when we're looking to understand what's going on in a disease, or if a treatment works, if we can do this across multiple model systems, then that really adds more weight to our scientific findings. And so one study in a single system doesn't really provide us with enough information to say that something is happening for sure. And so we need multiple different model systems, and we need those experiments to be repeated to understand whether this is actually what is happening in the cell. And so for that reason, building up these studies across different systems can take time, and that's one of the reasons why some of the steps in progressing some of this research can really take some time, because we've got to make sure that what we think is happening is actually happening. One really good way of understanding if these things that we're looking at in these transgenic systems are actually relevant to SCA8 disease and SCA8 patients is the use of biological samples from patients. And so one of these is patient-derived cell lines. So these might be derived from a skin punch or a skin biopsy. And so what would happen with that is we would take that skin biopsy and we would grow that in the lab. And that skin biopsy would generate a cell line that we would culture called fibroblasts. And so using patient-derived fibroblasts can tell us whether cells from patients from different patients have these disease proteins that we're interested in and what molecular pathways are affected in those cells. We can also use fibroblasts or cells from blood to actually generate neurons that we can study in the lab as well. And so those are referred to as iPSC-derived neurons. They're derived from turning the cells that we get from skin punches or blood back into a stem cell state where we can then move them forwards to be neurons. And with that, we can start to understand not only what's happening within the cell, but we can start to understand how those neurons are behaving and how they're firing or misfiring. And I'll come back to that a little bit later with some of the therapy studies. What we can also get from blood is DNA. And so looking at the DNA of SCA8 patients has actually taught us a lot about the reduced penetrance of the disease and what we can do to try and understand that. Another patient biological sample that's very useful is cerebrospinal fluid, or CSF. And so this is a fluid that really bathes your brain, keeps your brain and your spinal cord nice and healthy. And because of that, it contains a lot of the proteins and a lot of the RNAs that are in your nervous tissue, that are in your brain and your spinal cord, and we can measure those. And so CSF is a really powerful source for looking at fluid biomarkers from patients. So this is something that has been used so far more in, say, SCA1, SCA2, and SCA3. But down the line, it's something that could potentially also lead to looking at fluid biomarkers for SCA8. And then finally, something that's been really valuable for the study of Spinocerebellar Ataxia type 8 is actually autopsy tissue from the National Ataxia Foundation brain bank. And this really allows us to see that the proteins that we're looking at in our mouse model and in our transgenic cell systems are really relevant to disease because they're present in patient brains. And so that can tell us a lot about what happens during the disease process and can provide us with important therapeutic targets for looking at. And so I'm going to show you some data from that later on as well. Now, finally, I want to talk about natural history studies and how we can use those to study the disease. So typically, natural history studies will follow a group of patients over time. And so they will look to see when those individuals start to present with disease symptoms and follow as those disease symptoms develop over time. For SCA8, we don't have any formal natural history studies that have been published, that have been reported on, but we have learned a lot of information about SCA8 disease in patients and how it presents from case studies. So case studies are studies of maybe individual patients, a family of patients, or larger cohorts that are collected over time. But in general, these case studies don't tend to follow those patients over a long period of time. And so in the case of the pie chart that I'm showing you over here, this was a collection of 77 families that was built up over decades. And for most of those families, we only have a single time point that we've captured in looking at these individuals. And so one thing that we've learned from this case study is being able to understand how this disease presents in families. So earlier I mentioned the idea of reduced penetrance. So where multiple people in a family might have the repeat expansion, but maybe only one of those individuals actually develops SCA8 symptoms. And so that would be an example of reduced penetrance. And those families could be said to present sporadically. And so in this cohort of 77 families, that was actually the case for 63 of the families. So the vast majority of the SCA8 cases in this study actually only had one individual in the family that was affected by SCA8, despite in the cases where we did have more than just one individual, that repeat expansion would be present in multiple other individuals in the family. Of all the families studied, there were 10 families that presented with an autosomal dominant pattern of SCA8 disease. So that would be at least one affected parent and one affected child, and they would both carry the repeat expansion. And so this is what you would expect in a fully penetrant disease. And so being able to look at the differences between families that present dominantly and families that present with SCA in a more sporadic manner, we can start to understand some of the reasons behind the reduced penetrance. There was also a small subset of families, 4 families, that actually presented in a recessive manner. And so in a recessive disease, you typically need 2 copies of the mutation to develop disease. And what that generally looks like in a family is that you will have no affected parents, but you'll have multiple affected children. Now, in the case of SCA8, individuals only need 1 copy of the repeat expansion, but we see multiple families where one parent would carry the repeat expansion, but would not show symptoms, and multiple children would carry that 1 repeat expansion, but develop symptoms. And so this was a small group of the families that we found in this large cohort of families actually presented in that way. And typically, and I won't say that in all cases, but typically these families were actually primarily childhood onset cases of Spinocerebellar Ataxia type 8. What we've also learned from looking at case studies is understanding how repeat length relates to disease. And so this is why I said I didn't want you to think too much about those specific numbers that I used earlier to demonstrate the idea of a repeat expansion. What we have here are patients who are affected by SCA8. And so you will see that each dot here represents an individual, and the length of their repeat expansion is shown here. And so you'll see that the majority of these affected individuals do have repeat expansions that fall between that 70, 80 lower limit and about 250. But we do have patients that develop SCA8 symptoms with longer repeat expansions. And likewise, while you can see that maybe asymptomatic individuals, so people who carry the expansion but don't develop symptoms, may have slightly shorter repeat expansions, really actually there isn't a significant difference in this. And so understanding why these individuals don't develop disease or haven't developed disease at the time point where we have studied them, but these individuals have, is something that is really key to the field. And so if we were able to follow these SCA8 patients over time, as well as the asymptomatic carriers, it would help to understand why some people develop symptoms and others do not. And it would also help us to understand why, at what point in time some of these individuals are likely to develop symptoms. Because we have an age of onset for SCA8 that ranges from birth to 79, it's very hard to actually assign a lifetime risk for SCA8. So I'm now going to move on to discussing therapeutic approaches and clinical trials for Spinocerebellar Ataxia type 8. So there are a variety of different things that we can think about targeting or treating to try and improve disease symptoms in SCA8. So one of the things that we can think about is actually improving neuron function and brain health. So making those neurons that don't always fire quite right, fire how they should, behave more like normal control neurons. We can also think about how to actually slow or stop the disease process. And so to do this, we need to think about the cause of the disease, which is that repeat expansion. So we can reduce expression of the repeat expansion. We can also think about reducing the production of some of those disease proteins that I've mentioned earlier. So let's think first about how we can improve neuron function and brain health. And I've just pulled this table from the National Ataxia Foundation website. And so this lists most of the clinical trials that are active or recruiting or planned for some of the spinocerebellar ataxias. And what you'll notice is that SCA8 is only included in one of these clinical trials. And that's a clinical trial where multiple types of spinocerebellar ataxias are included. And so because spinocerebellar ataxia is quite a rare CTG, CAG repeat SCA, benefiting from therapies that can be used across multiple different diseases is going to be really key for some of the earlier therapeutics that may come out for this disease. So looking at therapeutic approaches that can be used across multiple spinocerebellar ataxias, we can think about therapies that improve neuron function. So here we have 2 clinical trials that use small molecules that have neuroprotective effect. So the trial that was in that table previously, and this is also from the National Ataxia Foundation website. The trial that was on the previous table is this one here, where we're looking at regulating glutamate with a compound called tririluzole. And there's another one here that looks at regulating neuronal firing. And so trials like this, where they take multiple types of spinocerebellar ataxias, will really help to move therapies forward for some of the rarer or less studied SCAs, such as spinocerebellar ataxia type 8. Now, we can also think about slowing or stopping the disease process. And so I just want to spend a minute talking about how we can reduce the production of disease proteins. So spinocerebellar ataxia has been a really interesting disease to study. And one of the reasons for that is that from understanding that we make those proteins from the ataxin-8 gene, a new form of disease-associated translation was actually found. And so this is something called RAN translation, or repeat-associated non-AUG translation. When we talk about translation in terms of what's happening in a cell, we're talking about the process through which proteins are made. So normally you will have your DNA, and that will generate RNA. And that RNA, or that transcript, is then read. And so when you read RNA, 3 letters make 1 amino acid. And so normally your RNA will have a AUG. So it'll have a combination of those 3 bases, which is essentially like having the word "the" at the start of a sentence. Okay, it tells you where you're going to start making that protein from. Now, in repeat expansion diseases, where you have these really long stretches of, say, CAGs or CUGs, the translation machinery can get a little bit confused. And so it doesn't always start reading C and A and G together. So if it did, we would have CAG, 1 CAG codes for a glutamine amino acid. And so we would end up with a polyglutamine protein if we read this as a CAG, CAG, CAG. When the translation machinery gets confused, what can happen is that it can read it as an AGC. So if it read it as an AGC, then AGC actually codes for a different amino acid called serine. And so reading as an AGC, this would generate a polyserine protein. And likewise, if we started at the G, when we read it as a GCA, this would make a polyalanine protein. And so actually, from the ataxin-8 gene, we don't just make this polyglutamine protein, which is what I showed you earlier. We actually make 3 different proteins that are unique to disease and not found in control individuals. And so these proteins we know to be toxic to cells. And so, as I mentioned, RAN translation was actually discovered in SCA8, but it's now known to occur in at least 9 different diseases. And so what I'm showing you here is control an SCA patient autopsy tissue. This is from the cerebellum. And you can see here that we have these dark red dots here. These are the polyserine proteins within the cerebellum. And so what you can see is that these are present in our SCA8 cerebellum, but not in the control cerebellum here, which looks really nice and healthy. When we look at the control cerebellum, you can also see that with this particular staining to look at something called myelin, it's much bluer. We have much more of that myelination. And so myelin is essentially the insulator around your neuron. It's the insulator around those electrical cables that run through your brain. And so when we lose that, our neurons don't function so well. And what we can also see here is that actually in SCA8, as well as our neurons not having that insulation how they should, the neurons are also starting to break up and be damaged. And this is something that we can see by looking at neurofilament H, where you can see that we have these big, thick bands of neurofilament, which indicate that there is damage to those neurons. And so here what we're seeing is these repeat proteins that aren't normally made in control brains being associated with some of the signs of general neurodegeneration and some of the things that could be contributing to that neuron dysfunction. Because we know that this novel translation process, this disease-specific form of translation, occurs across multiple different diseases, now that's another area that can lead to therapies that can be shared across multiple diseases and benefit for SCA8. And so if we can understand how these RAN proteins are made, then that can help us to identify how to slow or stop that disease process and how we can reduce the detrimental toxic effects of those repeat proteins. So now I want to talk about some of the other therapeutic approaches that are in very early stages in the preclinical work. And so one of these approaches is to actually reduce expression of the repeat expansion. And so this is something that I've been working on for the past 3 years in the context of CAG repeat expansion spinocerebellar ataxias. And so while this work isn't specific to spinocerebellar ataxia type 8, it's a small molecule approach that could be relevant to SCA8. And so what we did here was we developed a reporter cell line. So we integrated 2 bits of DNA into the cell's genome that aren't normally there. And one of these has a CAG60 repeat with a nanoluciferase protein, which you can see here lights up. And so because of this, we can measure how much of that protein is. And this protein has our polyglutamine chains attached to it. From our CAG0, or our control, we generate firefly luciferase, which is another protein that lights up in a slightly different way. And so what we can do with this reporter cell line that expresses both of these proteins is we can treat with small molecules. So we can use a library of compounds. And with that, what we can do is we can measure the luciferase and we can look for compounds that lead to a reduction in the amount of this nanoluciferase with the polyglutamine protein compared to the amount of the firefly protein, which doesn't have that polyglutamine protein. And so the idea is that eventually this might lead to the identification of small molecules that could be used to reduce expression or reduce the production of those toxic polyglutamine proteins. There's also the potential for disease-specific approaches for spinocerebellar ataxia type 8. And so one of these includes the use of antisense oligonucleotides. So these are little sequences of nucleic acid that would bind to the ataxin A mRNA. And this would signal for an enzyme to come along. And so this particular enzyme would then chew up this RNA, leading to degradation of that RNA. And so this again would lead to a reduction in the ataxin 8 expression. And so these kind of approaches that are still in very early stages could lead to things that would reduce the expression from those repeat expansions, and over time might yield compounds that could potentially progress through to clinic. Okay, and so now I'm going to talk about some of the other research aspects outside of therapy development that are really important for spinocerebellar ataxia type 8. And one of these is this idea of reduced penetrance. And so why do some people with the repeat expansion develop symptoms and others do not? Why is there such a wide range of age of onset? And there are reports in the literature of a variety of different disease symptoms other than those core aspects of the loss of balance and coordination, the alterations in eye movement and in speech that coexist in individuals who have the repeat expansion and ataxia. And so the question as to whether these variable symptoms are associated with SCA8 or whether they're co-occurring. And these questions all affect the diagnosis of spinocerebellar ataxia type 8. So from what Celeste was saying during when you were all coming online and where you're from, it seems like the majority of the participants here are from North America, where genetic testing for SCA8 is routinely offered, but that is not the case all over the world. So some countries don't routinely test for SCA8 because it can be very hard to actually provide information as to whether or not an individual is likely to develop the disease. And so understanding this aspect of reduced penetrance of SCA8 is actually really, really important for being able to provide better care for patients and being able to ensure that we can accurately diagnose this disease, provide appropriate genetic counselling, and stratify patients appropriately moving forward with clinical trials. And so the idea here is to understand what makes these individuals who have this autosomal dominant presentation of SCA8 different from these individuals who have very sporadic presentation of this disease with only one affected individual per family. So we're back here to our ataxin OS and our ataxin 8 genes, which sit on opposite strands of the DNA. And we have our CTG repeat in ataxin OS, which is a CAG repeat in ataxin 8. And so actually sequencing these repeat expansions can tell us whether this repeat here is a pure repeat or whether it actually has differences in those units of repeats. And so I'm just going to talk about repeats in different sequence interruptions in the context of the CAG repeat, but these would be present in both the CTG and the CAG repeat. So when we sequenced these repeats for this large cohort of 77 families, what we found was that there was a variety of different interruptions detected. And so the most frequent of these was TAG and the CGG interruption. And so where we saw the TAGs was between the CAG expansion and the short polymorphic stretch of TAGs, which is polymorphic in SCA8 patients as well as in the general population. And so quite often we would see a sort of stuttering between a TAG and a CAG and a TAG and a CAG before this transition into that full TAG stretch. Now, sometimes this is a longer series of stuttering and sometimes it was shorter. The other type of interruption that was detected very frequently were these CGG interruptions. And so these CGG interruptions would either sit in a little row, so you might have, say, 3 or 5 of those CGGs in between your CAG repeat. Or what happened in some cases is you would have a CAG, CGG, CAG, CGG, very similar to this TAG, CAG, but this could be anywhere within that CAG repeat stretch. And so what we actually found when we looked at in particular the CGG interruptions was we found that these were more frequent in families that had dominant SCA8 compared to families that had sporadic presentation of SCA8. And they were more frequent in SCA8 patients than in individuals who had the repeat expansion but didn't develop disease. Now, here I'm just talking about these CGG interruptions. The rest of these interruptions were really too rare for us to be able to understand whether they have an influence on whether an individual is more or less likely to develop a disease or have differences in the age of onset, let's say. So thinking about these CGG interruptions, why might these be associated with individuals who develop the disease more frequently than individuals who do not? And so we're going to talk about these repeat proteins again. And so what we found was that these CGG interrupted repeats actually resulted in more RAM proteins than pure repeats did. So if we take a pedigree here, so this is a family where we have one affected individual, but 3 people who carry a pure CAG repeat expansion. Now, this pure CAG repeat expansion encodes for the polyglutamine, polyserine, and polyalanine proteins that I've mentioned previously. And this leads to a certain amount of cellular toxicity or cell death. When we look at this family here, where we have 3 individuals that have the repeat expansion and they all develop symptoms of SCA8 disease, we can see that families like this are more likely to have these CGG interruptions. And when we look at the RAN proteins produced when these CGG interruptions are present, we can see that there's actually an increase in RAN protein production. And this also led to an increase in the cellular toxicity. So overall, these CGG interrupted repeats were more toxic to cells than the pure repeats. And so this suggests that there might be a biological basis for why these CGG interruptions were associated with increased penetrance. Now, I stress the word suggests because this work was all done in our transgenic cell-based systems. So it's very expensive and time-consuming to generate mice and to study them. And so doing this in mouse models wasn't possible. And we did not have patient samples that contained enough of these repeats versus these repeats to be able to look at the differences in patient cell lines or in other patient materials. And so while we saw this very, very clearly in our transgenic cell system, that's only a single model system. And so to really understand what these might be doing, we would need to look in more model systems than just this. So what does this actually mean for SCA8 patients? Well, together this really suggests a lifetime risk model for SCA8. So if you have a pure repeat expansion, so you have a pure CAG, the individuals may be at risk for developing SCA8 at some point during their life, but they may not. If individuals have these CGG interruptions within their repeat expansions, they may have an increased lifetime risk of developing SCA8. And so that's what we saw in this one study with this large cohort. We also suggested that individuals who have ataxia symptoms, they have the SCA8 repeat expansion, and they don't have repeat expansions for any of the other spinocerebellar ataxia genes, and likely have SCA8. And so while this is a suggestion of a model, because this has only been shown in a single study, and we've only looked at the biological basis of this in one type of sample, we really need further work here before we can actually use this in the clinic to be able to influence how we think about presentation and diagnosis of SCA8 disease. And if you are interested in learning a little bit more about this study, the National Ataxia Foundation through SCAsource has an article that came out, I believe, today on this publication. And so what I've talked to you about here is CGG interruptions and how they may lead to individuals being more likely to develop spinocerebellar ataxia type 8. But that's not the whole picture. So did we learn anything else from studying these 77 individuals and from looking through case reports from the literature? So one thing that we saw was that some SCA8 patients actually inherit ataxin 8OS and ataxin 8 repeat expansion mutations from both parents. So here we have an example of an individual who inherits one repeat expansion from one parent and one normal allele from the other parent. But some individuals inherit a repeat expansion from both parents. And in the literature and in the study of the 77 families, all individuals that had two of these repeat expansions developed spinocerebellar ataxia type 8. I am unaware of any reports of individuals who are asymptomatic and have two repeat expansions. And several of these individuals who had these expansions also had sequence interruptions in one or both of their repeats. And so this is a second way in which we think that differences in these repeat expansions might lead to individuals being more likely to develop disease. But again, that's not a complete picture. And so really, we're still trying to understand this reduced penetrance of SCA8. So more studies that sequence these expanded alleles will be able to tell us if other types of interruptions actually influence disease penetrance. And having some of these longer studies where we follow individuals over time and understand if they are likely to develop SCA8 at any point in their life when they have these different types of interruptions will also help us to understand this reduced penetrance more. Another aspect that can help us to understand why some individuals may develop the disease and why some individuals may not is looking at variations in our DNA that are outside of the repeat expansion. And so the genetic code that codes for each of us and as an individual is slightly different. And some of these slight differences may influence why some people are more likely to develop disease than others. And one way that we can look at this is through something called genome-wide association studies. And these can help to understand why some individuals may develop a disease earlier than other individuals. And so this is something that has been done for the CAG repeat expansion disease Huntington's and has really improved the understanding of Huntington's disease researchers as to why there's a variability in the age of onset with Huntington's disease. Now, while genome-wide association studies really need large patient populations, it's unlikely that there'll be a GWAS study in SCA8 in the near future, but due to similarity with other CAG repeat expansion diseases, we could learn insights into SCA8 from genome-wide association studies into other spinocerebellar ataxias, which could have similar mechanisms of understanding why there are variability in how these diseases present. And so finally, I just want to leave you with some of the areas where you can start to think about how to participate in research and drug development. So there is a rare disease patient registry and natural history study called the Coordination of Rare Diseases at Stanford, or CORDS. So this CORDS registry can be really important to put patients in contact with appropriate research institutes and researchers. As I said earlier on, some clinical trials do accept adult SCA8 patients. So for example, the tririluzole trial. Now I've specified adult here because none of the clinical trials that I've looked at so far the inclusion criteria for these trials are that the individuals have to be between 18 and 79. And so none of the things that we've talked about today would be in clinical trials for children at any point in the near future. And then finally, if more clinical trials do come out accepting adult SCA8 patients, some of the best places to learn about these are going to be the National Ataxia Foundation and clinicaltrials.gov. And so with that, I'd just like to thank you all very much for listening, and I will hand it back over to Celeste.
Thank you so much for a fantastic presentation. Topics like reduced penetrance and RAND translation can get very complicated very quickly. So thank you for providing us and explaining to us that nuance. Before we jump into some of the questions, just a reminder, if you do have a question for our speaker today, you can drop it in the Q&A window at the bottom of your screen. And we also have a couple of general questions that I'm going to start off answering so we can get into some of the more scientific focused ones. So first, yes, we've had a few people ask if there's going to be a recording of this presentation made public. Yes, it's going to be available in about a week on the National Ataxia Foundation website, so you can rewatch all of this. Additionally, just scrolling up through the questions, there's a few people who've asked questions about SYNE1 gene mutations. This causes a different form of ataxia called Spinocerebellar Ataxia recessive type 8. They have very similar acronyms, but they are different conditions with slightly different symptoms. We don't have a webinar series yet for SCAR8, but if you do have any questions, please feel free to reach out to the National Ataxia Foundation. You can get in touch with me at research@ataxia.org or celeste@ataxia.org if you have any questions in the meantime. And then we've had a few people ask, how can we get in touch or involved with natural history studies? The National Ataxia Foundation is actually involved in sponsoring one of the longest running natural history studies for ataxia called and this is a mouthful, the Clinical Research Consortium for the Study of Cerebellar Ataxia. And they recently started opening it up to folks who have a SCA8 diagnosis. So one of our staff members is going to put that link in the chat if you want to get in touch with one of the sites. It's available across the US, as well as there's certain sites in Canada as well. And then last but not least, we've had a few people ask, well, I want to get genetic testing. I don't know where to start. We have a resource page available through the National Ataxia Foundation that is also going to be put in the chat so you can explore some of the options available to you. So sorry, that was a mouthful. Let's jump into some of the more research-focused questions. So the first being, is there any understanding, or let me preface this. You talked about RAN toxic proteins occurring in the brain. Do we know if those happen anywhere else in the body? And if so, why do we only see symptoms related to the brain with things like balance and coordination?
That is a phenomenal question. And so while I think of an answer to that, I just want to say that it's fantastic to hear that there will be natural history studies for SCA8 patients. And I didn't know that before this webinar today, and so I'm absolutely thrilled to hear that that's going to be happening. In terms of RAN proteins in the brain versus different symptoms, a lot of that is really to do with the biology of the cells, so what those cells normally do, and also to do with the expression level of the repeat expansion in the different cell types. So if you have a gene that is switched on, it's expressed in all your cell types, those genes are generally referred to as something called housekeeping genes. So they are essential for the function of every single cell type. But not all genes are expressed in all cells. And so you might have cells in your, we call it periphery, everywhere outside of your central nervous system, that don't actually express ataxin 8. Now, the other thing that's really key here about neurons is that once you have your neurons don't divide and make more neurons. So those neurons accumulate all their damage over time. And so once the damage is too much, those neurons then die. And so if you have cells that divide, like your skin cells do, they divide and they die, but you will have new skin cells. They can split their rubbish up into every division that they make. And so that can keep on top of things, and it can help them to prevent the accumulation of damage, and those cells that contain a lot of those proteins might die, but it's not going to be problematic because you'll have new skin cells to take their place. Whereas with neurons, you can't make more. And so when that damage accumulates, that's really problematic. So we don't fully understand it, but those are some of the basic biology ideas that would begin to explain why we do see kind of selectivity in the types of cells that are affected by these diseases.
That's really cool to know. Then moving down the question list, we have someone, to your knowledge, is there a specific blood type that's more susceptible to folks who have SCA8? Is there any connection between the brain and the blood?
Wow, that's a really interesting question. And honestly, it's something I haven't really thought about before. We are starting to think, this is kind of quite off topic, we are starting to think a lot more about there being a gut-brain axis. And so things in your digestive system influencing your brain and vice versa. But I haven't really thought very much about blood type. And so there isn't, and I'm pretty confident in saying this because I read through all of the literature before I gave this talk, I haven't found anything in the literature that would link different blood types to SCA8, but I might be mistaken there. And so, yeah, I don't know. I don't know if it's been looked at.
So our next question involves some of what you were recently talking about at the end of your presentation about the CGG interruptions. Is that if someone, we have a couple folks here who have had different genetic testing, they haven't received any details about their repeat length with the CGG interruptions necessarily. Is that something fairly new? Is it only available in research context? How can people find that information?
So that's a really great question. So as far as I'm aware, clinical testing is still based on repeat length passing a specific threshold. So the study that I showed you data from is something that myself and others in Laura's lab did, where we sequenced patient DNA that we had in our bank. Now, that was just done from a research perspective at the moment. And so hopefully over time, there will be more studies that look at the repeat interruptions. And I don't know if that's something that's planned as part of the National Ataxia Foundation natural history study. But I think that hopefully over time that will make it to the clinic. But right now we don't have enough scientific data to be able to provide accurate genetic counseling based on that. So we do need
so it's So it's such a new finding and it's such a recent development that just hasn't had time to make its way out and people to implement it going forward.
No. Yeah.
Okay, that's good to know. Our next question is about the different therapeutic strategies that people are researching. Are you aware of anyone using gene editing or CRISPR approaches to test out for SCA8 treatment?
So not for SCA8. There are people exploring CRISPR to remove some of the repeat expansion for SCA1 and more generally for CAG, CTG repeats. These approaches are still very early and have very mixed results. And I will say it is a lot harder than you think to cut out a repeat. Repeats are slippy and tricky. And so it can be very hard to cut them down and keep them short. There is, however, a study using a small molecule that can prevent those repeats from continuing to expand and can actually shorten them in a disease, in Huntington's disease. So that could eventually have implications across CAG diseases, but right now, I don't know of any CRISPR studies in SCA8 now.
Well, room to grow, room to develop.
Room to develop.
Yeah, our next question is a clarification question about one of the charts you had in your presentation. So thank you so much. The chart with repeats showing affected versus non-affected individuals seem not to have too much significance to the disease. There's a lot of overlap there, and folks who had symptoms and folks who didn't have symptoms. How is it determined then that a repeat of any length is the source of SCA8 to begin with?
So this is a phenomenal question. This is something that researchers have debated since 1999 when the first study of SCA8 was published. And so that's why I'm just going to go back, if I can, to this lifetime risk model. So, you know, this is what we suggested in the paper. And again, this is an individual study, so it won't make its way to clinic yet. It needs more data to support it. But really, if we start down here and we think about patients who have ataxia symptoms, they do not test positive for any of the other CAG ataxias or different types of ataxia, but they do have that this repeat expansion in their ataxin A2S, ataxin 8 genes, then you likely have SCA8. And we know that if we express that repeat in a mouse model, those mice develop very similar symptoms to ataxia patients. So they have a loss of balance, they can't move like normal mice do, and they also express a lot of the same proteins in the brains as we see in autopsy tissue. So it is clear that the repeat can lead to disease, but understanding what other factors influence why some people do get the disease and other people do not is something that is a really big question in the field, something that came up at the last conference we were both at. So.
Yes,
Yeah.
and then we have time for one last question on that topic specifically about what else is known that impacts when people start getting symptoms. Is there anything known outside of the genetics if there is different environmental factors, as we call them within research, such as stress, trauma, injury? Is there anything known about that in connection to SCA8?
There is nothing published, nothing concrete. And I think that this is where those natural history studies are going to be really important. So understanding more about an individual's DNA in terms of the variation of the sequence of the repeat and the other genetic factors that could influence it, but also understanding more about those individuals' lifestyles. Did they used to be really active? Do they have increased stress in their life? And things like that will help us to understand other factors extraneous to the DNA that could influence whether people develop the disease.
That's really good to know. So another reason that natural history study participation gives us a lot more information when you have the numbers of different, higher number of patients participating. And with that, we are at the top of the hour. So unfortunately, we've run out of time. If we didn't get to your question, my apologies. If you'd like to reach out to research@ataxia.org, please feel free to send us an email. We can help you out the best you can. And another reminder, the recordings of this presentation and the presentation from Wednesday are going to be made available on the National Ataxia website sometime next week. And if we could all join me at our individual computers, thanking Dr. Shorrock so much for giving a fantastic presentation and the overview of SCA8. We truly appreciate it. So thank you so much for your time.
Thank you very much for having me. It's been, wonderful. And hopefully I've been able to answer some questions and provide some insight into where we're currently at with the research of this disease.
100%. And thanks to you, everyone, for joining today. Again, I hope you found something valuable and learned something today. And wherever you're joining us from, good morning, good afternoon, good evening. Hope you have a wonderful rest of your day. Bye, everyone.